Hello, this is Mrs. Schramm, and I am here to tell you guys a little bit about the Amgen Biotech experience. This is a lab series that we're going to be doing in class, and it's going to support all the stuff that we've been learning about. For example, the main thing that we know so far in biology is that the central dogma of biology is that DNA becomes RNA, becomes proteins. This process here of DNA becoming RNA, shown in this picture with the double helix being ripped open and then an RNA copy being made, that's the process of transcription that we've learned about in class. And then we have the process of translation, which is that RNA leaving the nucleus of our cells, finding a ribosome, and having tRNA assemble amino acids in the correct order to make sense of that DNA message. So we've seen that we have the DNA for hundreds and hundreds of different proteins in our body. One example of a protein in our body is this one right here. This complex down here is a chain of amino acids that we call melanin. Melanin is our skin color, helps absorb UV light, preventing mutations like skin cancer. But we have lots of other proteins besides melanin. So besides melanin, we have things such as this. This is hemoglobin, molecule responsible for bringing oxygen from our lungs to our entire body and delivering it to our cells. We also have complex, very complex protein structures, also made of chains of amino acids, but things that make little micro machines like this. This is an ATP synthase complex. Right here we've got tons of little amino acids that are making up each of these portions of the ATP synthase complex. So as you can see, but from subtle things like melanin that we can see to more complex things like hemoglobin or ATP synthase, proteins truly make up all of the functions and structures of our body. Now, the cool thing about these Amgen labs is that they support this idea that we've been learning about in biology class. So what these labs do is they provide us with a way to create a protein that some people are lacking. So you guys may be familiar with insulin. People with type 1 diabetes often have to take insulin because their body isn't making insulin. So for some reason, maybe it's like an autoimmune situation where their immune system has killed the beta cells of their pancreas, which are supposed to make insulin, so they're not making a sufficient amount to survive. They're going to need to take supplemental insulin. That's a protein that they're not making, that they need to make in order to live a healthy, normal life. These Amgen labs show us how we can take a couple steps back, go straight to the source of DNA, and use a human DNA gene for insulin and create that very human protein so that people with problems such as type 1 diabetes can have that pharmaceutical to use to live a normal life. This lab series is really cool because you guys are going to be learning how to create a small plasmid of DNA. Now, you guys have heard of chromosomes before. I've drawn chromosomes for you a lot. Here's a regular chromosome. Here's a chromosome multiplied into sister chromatids. But bacteria cells, which we're going to be using in this lab, they also, instead of using big chromosomes like humans have, they often share little tiny plasmids of DNA with each other. So what we can do is we can genetically engineer small plasmids of DNA. So let's erase a little bit of this. Okay? This circle right here is a double helix of DNA. So I could kind of draw, okay, this is a little double helix of DNA right here, this whole thing. And it can contain different genes that we're interested in. Right here, for example, I'm going to say that this gene represents the gene for human insulin. We're going to learn in these labs how to cut genes out using tiny micro scissors called restriction enzymes. We're also going to learn how to glue genes back together and stick them together using a covalent bond between that sugar phosphate backbone that we know, and create a plasmid that we can then put into a living bacteria cell. That living bacteria cell then will become a tiny, basically, protein-making machine that is going to make that insulin for us. It'll make tons of insulin and then we can take that insulin, purify it, and give it to people who need insulin. So that's what the lab series is going to be going over. And if I kind of show you guys what this actually is doing, instead of making insulin, which would be super cool to make in our lab, but insulin is actually clear like water, so it's not that exciting to see. We're going to be using something called the RFP gene, which is actually a red fluorescent protein found in a sea anemone. So we're going to be using that in place of the insulin gene to represent any gene of interest that we might want to put here to help somebody in need of some medical protein to live a healthy, normal life.